Hello everyone and welcome to today's webinar titled Dynamic Live Cell Imaging of Adherent and Immune Cells Using CellASIC ONIX2 Microfluidic Platform. This webinar is part of the Cell Bio 2021 and I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by MilliporeSigma. For more information about MilliporeSigma, go to www.sigmaaltrich.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate. First, by communicating with our attendees using our new live chat feature during the presentation. You can find this live chat located at the right-hand side of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as you have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on that Help Desk button located at the bottom of your screen with the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Jun Park, a senior scientist at MilliporeSigma. For a complete biography of Dr. Park, please visit the Biography tab at the top of left of your screen. Welcome, Dr. Park. You may now begin your presentation, sir.
Hello, my name is Jun Park, and I'm one of the R&D managers at MilliporeSigma. Today I would like to present dynamic live cell imaging of adherent and immune cells using our CellASIC platform. Here's the outline of today's presentation. I'll start with the basics and challenges associated with setting up dynamic live cell imaging. I will then give a brief intro to our turnkey CellASIC platform, which enables dynamic live cell imaging with any existing inverted microscopes. Main part of the presentation today will be applications demonstrating the utility of the CellASIC system with particular emphasis given to suspension cells such as T lymphocytes, which play a crucial role in immuno-oncology. Let's start with some background information on dynamic live cell imaging. I think by now it is very well established that live cell imaging offers tremendous biological insight over traditional static imaging due to its spatial and temporal information. For example, here we are showing the static image of T lymphocytes loaded with a fluorescent calcium indicator dye. At this point, not much insight can be gained, perhaps other than what appears to be different level of fluorescence among the T cells. Now, if we were to take time-lapse image of these T cells, as shown in the bottom of the slide, we start to gain deeper level of understanding to the spatiotemporal dynamics of these cells at single cell level. In this particular experiment, T cells were activated with ionomycin, which causes increase in intracellular calcium level. Already a couple observations can be made which were not possible when we're just looking at the static image in the middle of the slide. For example, we can notice that T cells are oscillating in their calcium level during the resting stage, and also that there is a significant level of heterogeneity among the T cells in terms of their calcium responses upon activation. Let's play the movie itself to better appreciate what is going on. In summary, the advantages of dynamic live cell imaging can be summarized in this slide. Analysis of cellular event at single cell level, thus enabling study of population heterogeneity. Study of cellular biology as a function of microenvironmental perturbations. For example, we can study calcium responses of T cells before, during, and after their activation. Another common example would be dynamic gas control, such as cycling between normoxic and hypoxic conditions. Last but not least is the time dimension, where we can now study various cellular responses, such as morphology, movement, signaling, as well as the cell-cell interaction, as a function of time to obtain spatiotemporal information of any given biological system. Now, let's take a step back and think about some of the challenges in terms of building or buying a system which will enable dynamic live cell imaging. Dynamic live cell imaging, while it is a very powerful tool, has its challenges. This is an image from Wikipedia and nicely demonstrates the complexity of such a system. These are very large and complex systems with a high price tag, requiring well-trained scientists to operate. Also, in all cases, additional do-it-yourself components are required to enable perfusion and fluidic capabilities in order to create true dynamic imaging system. These components, such as pumps and perfusion chambers with associated controllers, will further create complexity and increase price. Switching gears then, let's talk about our CellASIC platform. What would be the key advantages of our system? To start, it is a true turnkey product with integrated environmental control system coupled to microfluidic plate where cells will be cultured and imaged. The system controller, shown in the middle of the slide, is the size of a typical 13-inch laptop in its X and Y dimension. It is extremely portable and was built with mobility in mind. The controller controls various parameters such as temperature, gas, and flow rate within the microfluidics plate, which is connected to the controller by manifold. The software is very easy to use and enables step-by-step protocol building where each experimental step can be built with user-defined time, temperature, gas, and perfusion parameter such as flow rate. Let's take a look at the plate in greater details. It has standard 96 well footprint. Images in the center show one of our plate preloaded with colored dyes to show individual layout of the fluidics. Plate has 6 inlet wells on the left side and 2 outlet wells on the right side connected to the central tissue culture chamber area via microfluidics channel. As you can see in this cartoon, for each row, indicated as a group 1, 2, 3, and 4, all the individual wells within the group are connected to tissue culture chamber area via individual microfluidics channels, which are the colored lines. This enables up to 4 independent experiments to be conducted per plate. When the plate is connected to the controller unit via manifold, media or reagent in each of the inlet wells can be pressed down via positive pressure. This will then move the media or reagent from that well into the tissue culture chamber area, and which will exit to the outer wells. Essentially, this creates a system where any desired media or reagent can be precisely delivered to cells in the chamber area at desired time, for specific duration, and with specific flow rate. Since the plate can be temperature and gas controlled, we now have a system that can offer ability to control cellular microenvironment in dynamic, on-demand fashion. Plate material is also made of gas-permeable polymer to allow dynamic gas control. And the bottom of the plate is made of N1.5 glass for clear imaging, including confocal applications. Last thing I would like to mention is that since these are microfluidics plate, we can also introduce specific micro-dimensional features targeting certain applications, such as imaging of suspension cells, or maintenance of chemotactic gradient. Now let's go over some of the key applications. Our system can be connected to two independent gas outlets. So in this example, we can create normoxic and hypoxic conditions using gas switching ability. The middle image shows fluorescent intensity of reverse oxygen sensor, RTDP, measured from one of the tissue culture chamber within our microfluidic plate. RTDP will increase fluorescence with decreasing oxygen concentration and vice versa. Arrows indicate the time point at which gas switching was initiated. We can see RTDP fluorescence increasing as we put tissue chamber under hypoxic condition starting at T equals 2 hours. When we revert to normoxic condition at T equals 6.5 hours, we can see fluorescence start to decrease. Now that we have established that our system can create hypoxic or normoxic microenvironment, the image on the right shows cellular responses under such conditions by measuring expression level of HIF1 alpha, which is a key transcription factor under cellular hypoxia. We are also measuring RTDP fluorescence as indicated by blue line. When cells are subjected to hypoxic condition, we see the expression level of HIF1 alpha increasing as seen by the increase in the black line. And then later on, as cells are subjected to normoxic condition, we see fluorescence of RTDP decreasing, which in turn is followed rapidly by decreased expression of HIF1 alpha. Intensity of DAPI is plotted as a control in purple line, and it shows no meaningful change under changing oxygen levels. Next, we would like to show you live cell movie generated using BioTracker hypoxia live cell dye. This is a cell-permeable live cell dye, which becomes fluorescent under hypoxic condition inside of cells. Here we're looking at A431 cells loaded with and without dye after 16-hour incubation under hypoxic condition to show its specificity. Now let's take a look at the live cell movie. Next, we will look at the autophagosomes formation under hypoxia using fluorescent lysosomal marker LC3. Under hypoxic condition, cellular stress will induce the formation of punctate autophagosomes, and this can be visualized and measured based on live cell imaging. Let's take a look at the movie. Next applications, we're looking at intracellular trafficking of proteins. In this case, we are examining internalization of fluorescent EGF receptor upon EGF activation. You will notice upon the addition of EGF, the receptors start to coalesce together, which then gets internalized to endosomal compartment with characteristic punctate patterns. Let's take a look at the live cell movie. While the migration and invasion is widely performed on membrane platforms, they do not allow live cell imaging for obvious reasons. Here we now show how microfluidics can enable live cell imaging of cellular migration while maintaining chemotactic gradient. We have a microfluidics culture chamber that is connected to two parallel flow channels. Cells are cultured in the middle chamber, and chemoattractant is perfused into an adjacent channel. As you can see in the images at the bottom, cells will migrate towards the presence of chemoattractant. Let's take a look. Perhaps most obvious utility of dynamic live cell imaging is calcium imaging. Many cell types respond to external stimulation by increasing intracellular calcium level. Often these calcium responses are transient responses which only last for short duration of time. Bottom images of the slide show calcium responses of HeLa cells to ATP, and you can see that the response occurs within a minute of ATP introduction. Imaging this type of rapid transient responses require on-demand dynamic imaging capability. Now let's take a closer look at the ATP-induced calcium responses of HeLa cells. We'll cover one more example of calcium imaging, where cells are undergoing rapid, spontaneous calcium oscillation. Image on the right shows cardiac muscle cells undergoing a spontaneous contraction. With live cell imaging, we can better understand the temporal dynamics of such behavior. Images on the left show calcium fluctuation of individual cells over time. We can see that the cells are actually beating every 2 to 3 seconds, as seen by the ups and downs of the calcium level. Let's take a closer look at the live cell movie. Next, I would like to cover live cell imaging of suspension cells, which pose unique challenge due to their non-adherent nature. As you can see on the image at the bottom left, during live cell imaging, suspension cells will move up and down, making it impossible to image cells at single cell level. However, if we introduce micron-scale sealing, then we can confine these cells under single focal plane and enable clear visualization of cells during live cell imaging. Now we can use this micron sealing feature to examine in great detail cell-cell interaction of T cells with that of antigen-presenting B cells. Let's take a closer look. Here's another example where we're examining T-cell apoptosis at single-cell level. We're using CD95 activation to induce programmed cell death, and then visualizing the cellular events using caspase-3 specific apoptosis probe, as well as annexin 5. Let's take a look at the live cell movie. I would like to close the application part with 3D live cell imaging. We know that currently 3D tissue culturing is a very hot topic. Shown on the left side are images of MCF cells grown in our microfluidic plate compared to a regular chamber slide. Let's take a closer look at how these cells form 3D structures in our plate. In closing out, I would like to mention some additional features of our system. In addition to dynamic live cell imaging, it can be used as an automation platform for tasks such as staining. We can also enable extraction of cells after imaging. For example, trypsinized cells can be interrogated by flow cytometry. Proteins or nucleic acid can also be collected and analyzed as well. That covers our applications overview. Now for summary, I would like to quickly go over our plate offerings. We have a number of plates targeted for different cell types. So all our plates that start with letter M are intended for mammalian imaging, whereas our plates with letter B and Y are intended for bacteria and yeast imaging respectively. We do enjoy a tremendous number of publications, many of them in top journals, further demonstrating the value that we bring for our customers. Now, in summary, we know that the real in vivo biology is complex and dynamic. An ability to perform dynamic live cell imaging offers a unique window of opportunity to better understand these events. With CellASIC, we can now better control the dynamic cellular microenvironment. With turnkey offering leveraging microfluidics, our system enables a wide range of studies which can better recapitulate complex in vivo biology. With that, I would like to thank everyone. If you have any questions, please do not hesitate to contact us or visit us at www.cellasic.com. Thank you.